everyone. Thank you for staying till the end. As Vicki said, my name is Stephanie Hicks. I am going to be talking a little bit about my own research in the fields of single-cell and spatial transcriptomics or gene expression data. So it's going to be a little bit different than what we've been talking about thus far today, but hopefully still exciting for you nonetheless. And then I'll talk a little bit at the end about open challenges and opportunities for translating this into the clinic. Okay, so in terms of healthcare research, one question that we often ask is, why do patients not respond to a particular drug treatment? Of course, we all know that there is heterogeneity between all of us here in the room. There's heterogeneity between the individual cells and our individual tissues. And identifying cell type specific biomarkers can really play a very important role in personalized medicine. So one of the things that I have been focused on is if we need to identify cell types, if we need to identify cell type specific biomarkers, how can we go about identifying just cell types themselves with genomic data that's of course accurate and fast? So the caveat here is there could probably be a different answer. Like if I asked each one of us in the room today, what is a cell type? How would you define a cell type? We probably would come up with our own different answer. So just keep that caveat in mind. But the goal here is to develop computational tools that allow us to use genomic data and identify cell types so that we can then go forward and identify cell type specific biomarkers and thinking about them in the context of personalized medicine. So for those that are not familiar with gene expression, I'm going to start with an analogy that I like with Legos. So I have 3 young kids. We have a lot of Legos in my house. So starting at the top is a 3D Lego of the human brain. And if we think about through time how we have been able to profile the human brain, for example, using gene expression technologies, starting on the left is bulk RNA sequencing. So thinking about it as like a big pile of Legos, whenever we want to, for example, use bulk RNA sequencing, what we're really getting is an average of expression across all the cells in that tissue. So the analogy for bulk RNA-seq is it's like we get to measure the width of all the Legos, for example, or the height or the depth of all the Legos. Fast forward to 2014, 2015, when single-cell technologies came online. This is kind of like being able to take our big pile of Legos and start to separate them into little piles of Legos based on color, shape, or size. We get a rough idea of the types of cells in our data. We get a rough idea of the relative fraction of cells in our data, and so forth. And then the final technology, really like the current technology that we're using to profile these data are what's called spatially resolved transcriptomics. So I'm going to focus on the latter two for most of today. In spatial transcriptomics, the analogy really is thinking about putting these Legos together in a 2D space so you can see where the genes are expressed across the 2D domains. And I will say that the technologies themselves of spatially resolved transcriptomics, they were labeled by Nature Methods in 2020 as the method of the year. So these are really robust technologies that are hitting their stride and being used more and more in just basic research labs. I would say not so much in the clinic yet, and I'll get to kind of why. Okay, so what can we do with these data? So if we think about the brain, for example, a lot of what we know from the brain comes from either GWAS studies, like we had a presentation on earlier today, or from bulk gene expression papers, where we either get some kind of GWAS hits, TWAS hits, rare variants, or maybe we have a set of genes that we curate from bulk RNA-seq studies that compared, for example, neurotypical and schizophrenia patients. And the idea is we want to figure out how the genes that are enriched, for example, from the bulk RNA-seq study are enriched in maybe a particular cell type or maybe in a particular domain, a spatial domain in 2D space. So there are 3 parts to my talk. I'm first going to give kind of a brief overview of genomic data and my research interest with genomic data. I was told that this could be a particularly broad audience, including clinicians who might not know anything about genomics, so I wanted to briefly touch on that. I'll give 2 examples of my work that's really been focused on developing computational methods for the analysis of single-cell and spatial data. And then at the end, I'll wrap up with some kind of forward-thinking thoughts about challenges and opportunities for genomics and healthcare. Okay, so a little bit about genomics for anybody who is new to this. If we think about the wonderful talks that we had this morning, and we think about what are genomic data, at its core, what often happens, and this is, I would say, harkening back to short read sequencing, not necessarily long read sequencing. So this is like, I guess, like an older way that we might profile genomic data, but bear with me. So imagine we have a piece of DNA. If we wanted to generate some genomic data, what we would do is we would typically go through a set of experiments. These DNA would get fragmented, and we generate what's called a set of sequencing reads. These reads in the world of short read sequencing can be like 50 to 200 base pairs in length. When you put all these reads together in what's called a FASTQ file, these files can be quite big and quite illegible to anybody with like a human eye, but there is a lot of method to the madness, and there's a lot of structure in this data. So what do we do with all of these reads? If we think about having just maybe a reference genome, let's all agree that we do have a reference genome of some kind, and we have a gene of interest, gene XYZ, labeled in yellow. If we have a set of sequencing reads that comes off of our sequencing experiment from that FASTQ file that I just showed you, we have to figure out where it maps to best based against some kind of reference genome. And here, each read is represented by a black line, and I'm showing you the best pairwise alignment, for example, in this, against this reference genome. And at its core, what we often do when we talk about quantification is try to essentially sum up those reads for a given gene. And in this case, there are 23 reads that are mapping best to gene XYZ, and then we record that number in a matrix. And this is the matrix that I'll explain here in a second that I often use in my downstream analyses. So again, 23 represents 23 reads that map best to gene 1 in sample 1. We repeat this process for all the samples in our data. So for example, in sample 2, there were only 10 reads mapping to gene 1. We repeat this process for all the genes in our data set. So this is what I like to refer to as the counts matrix, and the matrix that I will talk about mostly today. But in addition to this, there's another matrix that we often use in genomics. It's a very important matrix. I like to think about it as important bookkeeping. Essentially, what is the gene's location, for example? What chromosome does it come from? What's the base pair position it starts at? What's the base pair position it ends at? And then this is, again, the data that we would typically analyze, the counts matrix, and we can ask some cool things, such as, let's take the average expression across 3 samples for gene 1, and then let's compare that to the average expression in the last 3 samples. And we can ask, is there a difference in the average expression between those sets of samples, for example? And we can repeat this process for all the genes, and we can ask which genes are most informative for example, differentiating between these 6 samples. Okay, so single cell. As I mentioned, bulk RNA-seq lets you get a really cool resolution of all up to, for example, all the transcripts expressed in a given sample, but it is an average of expression across all cells in that tissue. Single-cell RNA-seq came online in 2014, 2015, and the idea is the following. Imagine you have a tissue, for example, a tumor tissue, a brain tissue. Single-cell RNA-seq is a process where you isolate and or disassociate and sequence each individual cell, shown by the little cells here. I don't know if the mouse works. And then for each individual cell, you get a molecular readout of all the genes expressed for a given cell. That's what's shown in the top right with those little bars. There are 18 bars, 18 little black lines that map best to gene 1 in cell 1 here, and then that number gets recorded in a matrix. This is the count matrix that I talked about before. We repeat this process for all the cells and all of the genes and the data that we're profiling. One thing you should know about this matrix, though, is that in single-cell RNA-seq, this matrix is very sparse compared to bulk RNA-seq. So in bulk RNA-seq, this matrix may contain like less than 20% zeros, but in single-cell, it can be anywhere between 50% and 99% zero. So it's very, very sparse. Nonetheless, you can do a lot of great things with it. You can, of course, plot your data. You can plot the genes that are expressed in certain cells and see differences, or you can take our very high-dimensional data. So for example, if I'm profiling human data, we have on average around 20,000 human protein coding genes. We can take those 20,000 features and project them into a lower dimensional space using something like principal components analysis and ask how the cells separate in a 2D space. So in my principal components plot there on the right, you can see I have two different types of cells. Oh, hi. You have a question?
So what's the colors of the cells?
What is the color what?
The isolate and the sequence individual cells. What's the colors?
Green and red. Oh, the colors right here?
No. Second top.
This one?
On the top second. Isolate and the sequence individual cells.
So the colors are blue, red, green, and gray. So when we apply principal components analysis to single cell space, what that often does is reduces noise, and it allows us to work with a smaller data set that's usually more computationally efficient for downstream analyses. And so here you can start to see cells separating out in a 2D space.
So how do you separate the cells? By the disease or by the gender? Or?
So the cells, I'm just coloring by cell type right now, and I haven't told you anything about how I separate them. I'm just showing you a cartoon of what one could do with these data.
You don't know the types of the cells. What types?
In this particular case, it's just a cartoon.
Okay, so then the single cell doesn't separate the types of the cells.
So whenever you do single-cell RNA sequencing, you typically don't have information about the cell type, and you use the data to help you discover what are the cell types in your data.
Thank you.
Great question. So my research is really focused on developing very fast, scalable computational tools, and I'll talk more about why that's really important, in open source software for the analysis of these data, for both single cell and spatial data. With cells being an example on the right, and an example of a 2D tissue section shown on, cells are on the left, and the 2D tissue section is on the right. So I'll give examples of both today. Okay, so first I'm going to talk about scalable tools for single cell. So this was brought up in one of the earlier talks today, the Human Cell Atlas, and who's heard of the Human Cell Atlas? We're a small crowd. Okay, awesome. So the Human Cell Atlas is a worldwide organization it has a goal to create a map of every cell in the human body. It's an incredible, incredible goal, and many labs across the world are like part of this mission. And in 2018 or 2019, they published a white paper stating that as an initial draft, as a first draft, they aim to have an atlas with over 10 billion cells or 10 billion observations. That's a pretty big data set if you start to think about it. Imagine a CSV file with 20,000 features and a billion cells, or a billion by 20,000, for example, or 10 billion, 10 billion. That's really big. You might be thinking, okay, that's like pathologically large. Like, what are some example sizes of data sets today? So I picked out 4 studies from, I think, 2020, maybe 2019 and 2020, where I'm showing you in red the number of cells that were sequenced as part of each one of these studies. And my point here is to show you that we are standardly producing data sets with 1 million cells. So if we go to develop computational tools to analyze these data, I want everyone in this room to know that if you develop an algorithm that doesn't scale to millions of cells, it will not be able to be used in practice. So it's very important to think about computationally scalable tools so that they can be used in practice. So one of the things that my lab has focused on is really thinking about how to take existing tools that work really well and make them scale. I'm going to dive into a project on unsupervised clustering. So unsupervised clustering is a standard tool that we use to computationally partition 10 billion observations or 10 billion cells into groups, and then we try to associate meaning or biological meaning to each one of those groups, and often that's associating it with some kind of cell type. So if we want to cluster, for example, millions of cells, what do we need? Well, first, you need to have an algorithm that works with data either in memory, like let's say we load in a CSV file into memory, or data that is stored entirely on disk. So the data are not read into memory, it's in its entirety at any given point in the algorithm. The idea is you load in a portion of the memory, you do some kind of calculation, you spit that out, and you traverse over the entire data set. A lot of tools today actually require data to be read into memory, and you have to think about clever ways of adopting existing tools to be able to make them work interchangeably with either one of these. If you go to interact with any of the data in the Human Cell Atlas data portal, these are the file formats that you will encounter. You won't find a CSV file, you'll find what's called an HDF5 file, a ZAR file, or a TileDB. These are file formats that store data on disk. So you will immediately find these file formats if you go to work with these large data sets. And then of course we want our tools to be fast. We don't want to wait for a week to be able to have our data partitioned into a set of groups. We may want to partition our data multiple times. So k-means is a classic algorithm that people use in all walks of science. But it has a user-defined parameter, k, that requires you to decide how many groups to partition your data into. So maybe you want to try out k equals 5, k equals 10, k equals 20, and something like that. So k-means. K-means is, again, a widely used algorithm. For anybody who's not familiar with it, I thought I would give a brief overview. The algorithm is the following. Given a set of n data points, so let's say you have 1 million cells, so n equals 1 million, in some kind of matrix X, and a number k. So k is user-defined, and it's just the number of groups you want to partition your 1 million cells into. What k-means does formally is it partitions those 1 million cells into k groups. So say, for example, I choose k equals 10. Formally, k-means aims at minimizing this within cluster sum of squares criterion, shown where X is, again, as our matrix, and we're interested in estimating mu I, which are centroids for each of the centers of the k profiles, for each of the I groups up to the number k. In practice, though, I will tell you, we don't actually do that. In practice, we use something called Lloyd's algorithm. It's the default algorithm, often in Python and R, that you will go to interact with if you load up k-means. Lloyd's algorithm is an iterative algorithm based on 2 steps. First is an assignment step. So it assumes you have some kind of set of centroids. It assumes mu I has already been set. And then what it does is it just says, what is the closest distance between each observation and its closest centroid? And it gives that the assigned label. So if my first observation is closest to my first centroid, then I get labeled as group 1, for example. Assume you now have assigned each observation to its closest centroid. Then what it does is it says, now I'm going to estimate a new centroid for all the observations in cluster 1, all the observations in cluster 2, and so forth. It just iterates back and forth. It works really well in practice. That being said, it does require a couple of things. 1, it requires all the data to be stored in memory, and 2, it requires you to store all those pairwise distances between each observation and its closest centroid in memory. And if you're working with millions of cells, that is a non-trivial matrix that you are storing in memory. So we set out to make this a little bit faster using something called mini-batch k-means. It's a super intuitive algorithm that is a natural candidate for clustering data on disk. So the idea is the following. If, let's say, you have your data stored on disk, you can load in 1% of the data into memory or 5% of the data into memory. They call that a mini batch, or the batch size. A batch size can be relative, such as 1% or 5%, or it can be absolute, such as 100 observations or 500 observations. You load in 1% of the data, you update your centroids, you spit that out of memory. You load in the next 1% of the data, you update your centroids, you spit that out of memory. You traverse over the entire data set. At the end, there's a final assignment step where you assign each observation to its closest centroid. And this is a very natural way to make clustering scalable for very large data. So one, you don't have to store the whole data into memory. You only store in the size of the data, the batch size that you want as a user. At each iteration, only the distances between each mini batch and its closest centroid need to be computed, and this makes it a very natural candidate for on-disk clustering. So there were some existing implementations at the time. Yes? The question is, do I see batch effects? So the question is, do I see batch effects? So yes, there are tons of batch effects in single-cell RNA-seq data. That is a totally, in my opinion, like a topic that warrants much discussion and debate that I'm not really touching on here today, but it absolutely would show, if you had batch effects in your data and you had not corrected for them, you would see them here. Good question. So there were some existing implementations at the time. One was in R, in the cluster R package, and if you wanted to use the file formats that I talked about before, HDF5, TileDB, and ZAR, it cannot work. It only allows you to have the data stored in memory. So this is a problem. And then there is an existing implementation in scikit-learn. Here, the data can be stored in an HDF5 file. That was really exciting. But if you go to execute the algorithm, there's like a step at the very beginning, and what it does is it just reads the whole data set right into memory, and then you're off and running. So that one was like not so desirable. So we implemented our own version called mini batch k-means. It's designed to be a simple replacement to k-means just to make it scalable. So anywhere you have a data set with lots of observations that you want to partition into k groups, you just simply replace k-means with mini batch k-means. You have to define a batch size, but you're off and running. So it's really meant to be just a scalable version of k-means. But this was motivated by single cell in the world where we are generating these very large atlases and we want to cluster our data very fast. So how does it work? Well, the take-home message here is that mini batch k-means is very memory efficient and very fast. So we took a data set with 1.3 million mouse neurons and we downsampled to smaller sizes of data. So on the left plot, on the x-axis, I'm showing you the size of the data set, or the number of cells in our data set, ranging from 75,000 up to 1 million on the x-axis. And on the y-axis, I'm showing you the max memory used over the course of that algorithm. And we profiled 3 different algorithms. So one is Lloyd's algorithm, base k-means, shown in red. And then, let's see if I can, right here. And then in the one in green is mini batch k-means with the data read in memory. So let's say you refuse to work with data on disk, you have a CSV file, you have a very large cluster, your cluster can read in a lot of data. That's the green, that's the performance with the green line. And then the blue line shown down here is if you have your data stored on disk in an HDF5 file using mini batch k-means. So what you can see on the left side is that k-means is very memory inefficient. The amount of memory resources that you need to request becomes quite big as your data become quite big. Oh, and I forgot to mention, the plot on the right is the same thing except we profiled the amount of time it took to complete each algorithm. So k-means is very memory inefficient and can be quite slow as the data become larger, above a couple 100,000. And then mini batch k-means, you get huge cost savings. Even if you don't want to deal with an HDF5 file, you get huge cost savings in terms of memory efficiency and speed. And then if you are willing to work with an HDF5 file shown in blue, you can cluster a million cells in less than 2 gigabytes of memory and under 10 minutes, which is quite phenomenal. The first time I saw that, I was really blown away. What's the catch? There's always a catch whenever you try to make something faster. The take-home message here is that mini-batch k-means is as accurate as k-means for a large enough batch size. So I've given you kind of this intuition about batch size, 1% of the data, 5% of the data, or 100 cells or 500 cells. I'm showing you now 3 different sizes of data sets ranging from 5,000 on the left, 10,000 observations in the middle, 25,000 observations on the right. On the x-axis is the absolute batch size, so 200 cells, 500 cells, 750 cells. So this is the batch size I'm using as part of my algorithm in mini-batch k-means. And on the y-axis is the within cluster sum of squares criterion. So this is a criterion that we often use to measure accuracy. We can argue about whether it's the right criterion, but this is often a criterion used to measure the accuracy of k-means. What you can see is k-means is flat. It is not a function of batch size, as it should be. And then in blue and green, what you see is for smaller batch sizes, you just get simply noisier estimates. And then as you increase the batch size, say up to 500 observations, the performance of what you get from mini-batch k-means and k-means is quite similar. Underneath the hood, mini-batch k-means is doing stochastic gradient descent for anybody who's interested. This approach of using mini-batches can be used anywhere you're using stochastic gradient descent. You can do stochastic gradient descent on one observation at a time. It is very noisy. And so this idea of using 500 observations or 1,000 observations gives us incredible performance improvements in terms of memory efficiency and speed without much loss of accuracy. So the last plot I'll show you here is we took our 1.3 million cells, our mouse neurons, and we applied mini-batch k-means to it. I'm showing you a UMAP representation of the predictions for each observation, but instead of trying to plot 1 million dots in a plot, I'm using what's called a hex bin plot. So this is a really clever way to visualize predictions with a lot of observations, because there's a lot of overplotting that can happen when you plot 1 million points in a 2D space. So what I did is I just grid up this 2D space into a series of hex bins, and then I color the hex bin by the majority label for the prediction for that cell, for those cells in the hex bin. So that's what the predictions are here, and the package that you can use is called SEHex if you want to do this for yourself. So what did we do? We took our 1.3 million cells, we applied principal components analysis and got the top 50 principal components, and then we clustered our data 16 different times to try out different versions of K. That whole process took 3 minutes. In contrast, state-of-the-art in both Seurat and ScanPy in Bioconductor is this concept of graph-based clustering, either using a shared nearest neighbor graph followed by Louvain clustering, or BBKN followed by Leiden clustering. Those processes took 35 minutes and 48 minutes respectively, so really demonstrating the performance improvement with 1.3 million cells. And this, I will say, this blue line here will continue to stay flat as the data become larger, because you're thresholding based on the size of the mini batch. So that's really cool to see. Okay, so we published our work in PLOS Computational Biology. There's a Bioconductor package that's available if anybody's interested. And now I will move into the second part of part 2 in spatial genomics. Sound good? Okay, so I'm going to continue with this idea of developing computational tools that are scalable for genomics data, but I'm going to introduce now formally this thing I've been mentioning a couple times, spatially resolved transcriptomics. I'll introduce a data set that I'll kind of talk through this next portion of the talk, and then I'll talk about the methodology that we created to answer an important question. Okay, so the data set that I want to talk about today is a set of experiments that were performed in postmortem human brain, shown here with this particular region, this slab of the postmortem human brain in the anterior region of the brain, of the dorsolateral prefrontal cortex. And we profiled the genes expressed in a 2D space from this particular region of the brain. Now imagine you took a slab of this brain region and you laid it in a 2D space. That's what this is. It was a slab from this 3D reconstruction of the brain laid in 2D space. And then this tiny little black box here is the region that we actually profiled with our spatial transcriptomics data. This particular region of the brain, the DLPFC, is very well studied in the literature. It has 6 layers that are within gray matter, so layers 1 through 6 right here, and I'll show you the layers in just a second. And then it has white matter, and you can kind of see it in its cartoon, layers 1 through 6, followed by white matter shown right there. So we picked this region of the brain because there is so much information that's already known about it. And if we saw something that we truly did not expect, then we could maybe attribute it to the technology not working as opposed to saying we've discovered something totally new in a region that has been so wildly studied. So the experimental design was the following. We had 3 postmortem human brains, so subject 1, 2, and 3. And I know we just had lunch, so bear with me. Imagine we have a 3D tissue block from this postmortem human brain. Imagine doing a deli slice and laying it in a 2D space. That is replicate 1 right there. Imagine doing a second consecutive deli slice. That is replicate 2. And then imagine moving into the tissue block 300 microns, deli slice 1 and deli slice 2 again. Does that make sense? So 2 deli slices, sorry for the terminology, moving into the block 300 microns, another 2 deli slices. So what you'll see is you'll get to see donor-to-donor variation, and then you'll get to see the variation that you get just doing 2 consecutive tissue sections. And a little bit about the technology, because it's helpful in the next part of the talk. The platform that we used, the experimental technology that we used, is from 10x Genomics Visium. The blue here shown, the blue here is the actual slide that you get, 1 Visium slide, and it can profile up to 4 tissue sections. They call these capture areas. If you zoom into 1 capture area, what you see is the following. On the outside of the capture area, you have something called the fiducial frame, shown in red. It's a set of spots that it lets you line up barcode-based sequencing data with an image. The image could be an H&E image, like a bright field image, or it could be an immunofluorescence image. But the fiducial frame is how you line up the barcode data with the image itself that you also get from a Visium technology. And then inside of the fiducial frame, you have up to 5,000 what are called spots. So each spot is essentially a set of barcodes and an XY coordinate plane that is that has a set of primers for a unique set of primers for each spot. So you have RNA that binds to the set of primers, for example, and when you go to sequence that RNA, you have a spatial barcode that tells you the unique location where that RNA came from. You have a UMI barcode, a unique molecular identifier that tells you which molecule these reads came from, and so on. So at the end of the day, you again get a counts matrix, except these counts, instead of cells, they come from these XY coordinate planes. And you have up to 5,000 observations per tissue section with up to 20,000 protein coding features. Okay, so let me give you a little taste of what this looks like. The first figure up here is the tissue that we profiled inside of the block itself before we put it on the Visium platform. And you can see just visually, well, I don't know if you can see it on the slide, this is darker than this area. The gray matter is darker than the white matter. And then I'm annotating what layers 1 through 6 will look like. And then here it is, the histology image, the bright field image shown with layers 1 through 6. And here you can see in the histology image, the white matter looks a little bit darker than the gray matter. And on the bottom row, starting on the left, is 3 genes, SNAP25, MOBP, and PCP4. So these genes are well-studied genes that are associated with each one of the layers. SNAP25 is a gene that's expressed everywhere there's gray matter. MOBP is unique to white matter. And then PCP4 is a gene that is beautifully expressing or associated with layer 5. It's associated with synaptic plasticity for anybody who's interested. So you can kind of get a sense of where this is going. So these genes are varying in space in a non-random way. They are associated with the morphology of this tissue, which is really cool to see. So zooming in on just one of those genes, PCP4, that's associated with layer 5, I want to note that in addition to the gene expression data that we have with this data set, there were 2 kind scientists who manually annotated each individual spot in these 12 tissue sections. So I'm showing you, I'm not going to get this right. Okay, I'm showing you the manual annotation for this tissue, and you can kind of see the purple and yellow light up where PCP4 is, if that makes sense. So you can kind of think about this as like a gold standard. So somebody has painstakingly manually annotated these layers based on what we know in the literature. And so whether or not you believe this is actually true, it's kind of useful for us in the sense that we get to see if what we see with the data-driven methods matches what we see with the manual annotations. Okay, so now I want to zoom out and show you the kind of donor-to-donor variation and tissue section to tissue section variation that you can get. So the design is the following. Along the rows are subject 1, subject 2, and subject 3. So these are the 3 different donors that I talked about. And then the first column is that first tissue section into the block, followed by the consecutive section right next to it. And then over here, these tissue sections are after you move into the block 300 microns. So a couple of things. One, you see huge donor-to-donor variation. There is a lot of variation across the donors, which is kind of expected. The folds in my brain are unique and to the folds in your brain, and like depending on where the pathologist dissected the brain, the folds themselves are kind of less interesting. But it is interesting from a computational perspective because let's say I have tissue sections from 10 neurotypical controls and 10 Alzheimer's donors, and I want to align them in Z-space for something, right? How do you align, for example, this with this with this? So it's a good question to think about how to deal with this morphological variation that's less interesting. And then also you can see, for example, here, this was these tissue sections, I forgot to mention, are 5.5 millimeters by 5.5 millimeters. They're very tiny. The tissue is always wet. It's always, a graduate student who has to put these very tiny tissue sections on these Visium slides. And so what often happens is like the tissue may break a little bit, right? And then you have to figure out how you handle that. It's not like you can immediately line these up perfectly just out of the box. Okay, so I have artificially and intentionally shown you a gene that's clearly varying in 2D space in a non-random way. I call this a spatially variable gene. I want to detect which genes are spatially varying in a 2D space with data alone. I don't want to have a biologist tell me. I want to do feature selection is the formal terminology. I want to figure out which genes are most important or varying in a 2D space in my tissue section of interest. So one of the things that I set out to work on is something called methods to identify spatially variable genes. Okay, so this is the same data that I've talked about before, and I want to highlight something interesting and different than just the morphology. So in the top row here, I'm showing you these same 3 genes that I've already shown you before, MOBP, PCP4, and SNAP25. These are genes that are associated with the layers or the morphology of this particular brain region. On the bottom, I'm showing you 3 new genes that are associated with immune or blood. And what I want you to see is that imagine, for example, there was a little blood vessel there. I don't actually get to see that. I don't have any protein that was like simultaneously tagged within this data, but these genes are also varying in a non-random way. They are spatially variable genes that were picked up by the algorithm that I'll talk about in just a second. But what I want to discover are genes that are maybe varying from morphology, from the layers, or maybe there's like a gradient of expression where it's high in the anterior region and it's low in the posterior region, for example. Or maybe these genes are associated with other types beyond just cell types, such as the blood vessels. Now, one other thing I want you to notice is that the 3 genes in the top, they have the amount of variation that you see is really like spread across the whole slide, while the genes in the bottom, the variation that you see is a little bit more spotty. That took us a long time to figure out, just staring at a bunch of plots. And so we like to think about this as like a bandwidth parameter. Other fields may refer to this in a different terminology, but I'm going to use the term bandwidth parameter or length scale in just a minute. But I want to just point out that different genes have different types of bandwidths. Okay, so feature selection in spatial data. We could, for example, try using a method that was developed for single cell. So the feature selection tool that's most widely used is something called highly variable gene selection. The idea is to basically fit what's called a mean variance relationship. So I'm showing you a plot with the empirical mean on the x-axis and the empirical variance on the y-axis, where every dot here is a gene. This blue line is fit to the mean variance relationship. This is well documented in the field of RNA sequencing. And the idea is to try to find a gene that's far away from this blue line. This blue line is what we expect from sampling. It's not interesting, but that particular gene might contain a lot of biological variation, more variation than what we would expect just under sampling. So that's non-spatial feature selection. We could try this and just treat each spot as a single cell. There's a long history of spatial statistics that was developed really for geospatial statistics, so like modeling mortality, for example. Moran's eye statistic is a classic approach that you could apply for feature selection. It's really just a measure of spatial autocorrelation. I won't say any more about it, but it's another way that we could rank genes by which genes have the most amount of spatial variation. And then there are some newer methods that were developed to detect spatially variable genes, and I'll talk about just one of them. So Gaussian processes have been around in geospatial statistics for quite some time. They are very slow, though, to fit in a per gene manner. So the idea here is we would fit these models for each individual gene for all the spots in our data, and we would then figure out which genes have the most amount of spatial variance using Gaussian process regression. Again, it is incredibly slow. It is actually cubic in time as a function of the number of spots. So it takes about like 30 minutes to fit each one of these models, and if you have 20,000 features, that's painfully slow. Nobody will sit through that. So there are a couple of ways to make this fast. One approach is to use what's called low rank models. Really, the idea here is just you're approximating the data at a smaller number of spatial coordinates. It has its pros and cons. And then an approach that we took was something called nearest neighbor Gaussian processes. It was introduced by someone named Abidata in 2016. It uses essentially what's called an approximate likelihood. You're just trying to approximate the full likelihood using a smaller set of nearest neighbors. To fit the models, it's linear as a function of the number of spots, and there are some existing implementations. So this is the approach we took, and we call it NNSVG. And the idea is, again, you fit a model for each individual gene. And the thing I want to highlight here is that we have this parameter L this is a gene-specific length parameter that you fit for each individual gene. So existing models, they just set this to be fixed, and they assume the gene length scale parameter is the same for every single gene, but I showed you before that the bandwidth or the length scale parameter actually varies quite a bit and is important for identifying spatially variable genes. We fit models with and without a spatial component, and then we estimate a likelihood ratio test. If you're interested in this, we estimate a likelihood ratio test to rank genes. So I want to show you these same 6 genes that I've shown you before. So on the top row, these are genes that are associated with the layers, and on the bottom row, they are genes that are associated with the immune and blood. What you can see here is that these 3 genes from the top all have a larger length scale parameter that was estimated by the Gaussian process model. And then the 3 genes in the bottom row over here, they all have a smaller length scale parameter. So this is a distribution of all of the estimated length scale parameters for all of the genes that we fit. And you can see that there really is a difference. And just to drive home, existing methods basically fix this at some particular value, and they assume genes all have the same bandwidth parameter. And we found this to be something incredibly important whenever you're fitting these models, that you need to have the flexibility of length scales to vary in a per gene manner. Okay, so how does it perform? So we went back to our same 6 genes here. These are the layer associated genes, and these are the genes that are associated with immune and blood. And what you can see is our approach, NNSVG, works really well, but also like highly variable genes works really well too, turns out, which was kind of a surprise to us, but also not really. So highly variable genes is just finding cell types. And it turns out when you do spatially variable gene selection, we are also discovering cell types. Now, the thing I want to point out though is if you ever want to find spatially variable genes beyond cell types, this is where spatially variable gene detection methods really can shine. So for example, this is a different data set from the mouse hippocampus that was measured on SlideSeq, and it has dots that were annotated by the particular cell type. And these are 2 genes. These are 2 genes that we found that vary in space. So it's more highly expressed in this region and it goes down, versus this gene was highly expressed in this region and then it goes down this way. And this is really, this is how spatially variable gene detection methods can really shine, that even within one particular cell type, you can see gradients of variation. So this was really cool. There's an implementation that exists in Bioconductor if anybody's interested. It's called NNSVG, and I will leave it there. Okay, so the last part of the talk that I want to highlight is something that we just got out. So I've talked a lot about these spatial data. Now, let's say I want to share these images with a collaborator, for example. I have generated the most beautiful images of the mouse hippocampus, and I want my collaborator to be able to go in and kind of annotate and play with the data. There are some existing tools that you can use to have a collaborator play with the data. These are all GUI-based, graphical user interfaces, GUI, meaning that you have to download a set of software. It's a specialized set of software. The 2 big ones are ImageJ or Fiji, and then Napari. These are the 2 big softwares that are available for you to start to play with these image data. But they are not desirable for several reasons. I may have a collaborator who doesn't know how to install Fiji or Napari, for example, or these software are not designed to open up multiple samples at the same time. They are also, they will often crash if you have, in a variety of different ways. I'm happy to talk more about this if people are interested. So what we did was we built something called Samui Browser. You can think about it as the Google Maps for images, for spatial transcriptomics. So the idea is the following. We took the TIFF files that come from the sequencing experiment, And we converted them into what are called GeoTIFF files. So these are the file formats that Google uses in Google Maps. And then what we did is we chunk the file up into tiny little chunks. And then we put it into an AWS bucket. And then what happens is that when you hit the browser, for example, on your phone or on your computer, just like in Google Maps, a small amount of data is downloaded from the web locally. And then as soon as you start to drag the image, more data are downloaded from the AWS bucket. And this is the idea for how to make it very, very fast. There's essentially no lag whenever you go to interact with the data. So again, the data are stored entirely in the browser. This is a screenshot of the browser. I'm going to try opening it up and showing it to you. And then let's see how this goes. Okay. I'll do Visium. We're doing a live demo. Don't ever do this. I'm just kidding. Okay, so this is a tissue section from the Visium platform. I'll turn off the gene expression and I'll show you the following. So you can see there's like essentially no lag in terms of being able to play with this data. Every dot here is a cell. So this is the nucleus of the cell colored by DAPI. And then if we zoom out, for example, I'll show you these. GFAP is a marker for astrocytes. NuIn is a marker for neurons. Oglio is a marker for oligodendrocytes. TMEM is a marker for microglia. So this is a way for your collaborator or anybody who's interested or a reviewer of your paper to like go in and see, for example, what cells are which ones. So like, for example, neurons and DAPI overlaying, that tells me that this is a neuron, for example. Or microglia, it's a big mess. Ogliodendrocytes, you can tell that this is an ogliodendrocyte and something like that. So also, because this is spatial transcriptomics, we get more than just immunofluorescence. So we get the following. So here are the 5,000 spots that are the up to 5,000 spots that you can capture on a Visium technology, for example. So you can like zoom in and you can see, okay, I have only one, looks like one cell in this particular spot. Well, I have, I don't know, 2 or 3 in this one, for example. So now you can also go in and, for example, say, I want to color by MOBP. I forgot to turn it back on. Okay, so that's MOBP. So you can see, for example, MOBP lights up. Can't do this with this computer. I don't have this. Okay, so anyways, hopefully you agree that this is a useful thing. It's like the Google Maps for spatial transcriptomics. It's a completely open source software, so you can create your own Amazon bucket and share a URL with a collaborator with your own data. Okay, and then the last 5 minutes, I want to give just kind of some forward-looking challenges and opportunities for how one might want to use these data in healthcare and in the clinic. So there have been quite a few reviews that I just wanted to highlight at the beginning of this section that have talked about how are we actually using these data right now. So this is a screenshot of a figure from this paper by Jin and Kim. Starting on the left side, you can see, I'm just going to do this. So starting on the left side, you can see that the section is thinking about profiling, for example, whole brain sections from mouse in particular, or developing brain or organoids or subregions. People are also thinking about behavioral experiments, so using spatial transcriptomics to assess responses to different types of stimuli. People are thinking about this in terms of neuronal projections. So what's the spatial transcriptomics that's happening in the same brain region or in a different brain region where neurons are projected into? And then they're also thinking about this in this example that I talked about, like let's say I have 10 neurotypical controls and 10 donors from a neuropsychiatric disorder or neurodevelopmental disorder. How is the spatial transcriptomics changing between these two groups? And I will say that these types of applications are being generated right now with spatial transcriptomics data, with the caveat that the tools to analyze these data are a little bit of the Wild West. Okay, and then one other thing I wanted to point out is that there are some design considerations whenever you go to use these data. So one thing that I often get asked is, why are you using Visium? Why are you not using a truly cellular technology? And my answer is, it depends on your question of interest. If you are interested in discovery, if you're interested in trying to figure out which genes are most important in my tissue of interest, you want the full transcriptome. You want to profile the full transcriptome. And so you want to consider sequencing-based technologies such as Visium or Cosmics and things like that. They're often not truly cellular. Like, you could see that there were multiple cells in a given spot. It's not truly cellular. But the idea is you get to capture the full transcriptome for your tissue of interest. So this is really discovery, hypothesis generation. Once you figure out your genes of interest, then the idea is now you often want to zoom in and look to see at a truly subcellular level what are the receptors and ligands that are being expressed between my two cells of interest. There, you really want to think about image-based technologies. So there are some design considerations that I would say are really important. In particular, when you get to the clinic, you may not be at the stage of hypothesis generating. You may be at the stage more of hypothesis testing and really validating something that's happening in the tissue of interest. There are a lot of computational challenges, I'll be honest. Sample processing remains a computational challenge. We're now thinking about doing long-read sequencing of single cell and spatial transcriptomics. So everything to date thus far is really short-read sequencing. And as you know, that misses out a lot of variation, as beautifully discussed by the earlier presenters today. There's a lot of effort in generating 3D spatial atlases, so taking consecutive sections of a particular tissue and aligning them together in a 3D space. People are thinking about spatial multi-omics, spatial temporal omics. It's really quite phenomenal, the pace of this field. And then if we want to think about the clinic itself, so this was an article written by Jasmine Plummer, who's the director of the Center for Spatial Omics at St. Jude's Research. She had this great article that I thought I would just try to summarize here in the last couple of minutes. So imagine walking into the doctor's office in search of some diagnosis that you have. And let's say a physician orders a test on taking a particular biopsy from you, and then orders a test to have that profiled on a spatial platform. And then the hospital's local core will be able to use this spatial profiling data as some kind of like diagnostic or precision medicine tool. The spatial data that you capture will be able to be used in the following way. Maybe we've already had a large enough atlas generated such that we know the disease subtypes, the spatial disease subtypes. And so when you take this biopsy, we'll be able to immediately figure out what the subtype is or what the diagnosis is. The next day, the physician delivers the diagnosis with the treatment plan. The whole process is quick and easy, highly effective, precision medicine, yay! Okay, so this is kind of like the dream, right? This is maybe the thing that we would like to have happen, but the reality is we are nowhere near that. Maybe, yes, but the road is challenging. So I just wanted to highlight some of my key takeaways for why this is so challenging. And for me, it's really this jump between basic research and the clinic. This is a huge jump. So as you can see, the spatial technologies are rapidly being developed and rapidly being adopted in basic research labs, which is really exciting. But then basic researchers often generate these atlases and they kind of leave their findings of this is really cool and new, they kind of like leave it at the door at the end of that. And so moving into the preclinical trial, the translational, like the clinical setting, it really remains much more unknown how to do that. But I agree that yes, this is likely to happen in the future. But again, there's a like a long way to go. How to bridge the technology and translational worlds. So, as I mentioned, to date, a lot of the spatial research has been focused on generating these large atlases. That's good. I mean, like, we have to have a baseline of what we expect. But the true power of spatial is really to be able to zoom in and ask questions about like how cells are communicating, or the interlay and interplay of signaling pathways in a 2D space. That's going to be like the true power horse and like the power of spatial profiling, in my opinion. Also, being able to get spatial signatures, so gene signatures that are spatially aware, or that you know in the context of where cells are in a 2D space. So like typical biopsies may only profile a very small percentage of this tissue sample, while spatial profiling may actually be able to capture a larger area of that particular tissue. And then, of course, we need more than gene maps, or gene lists and gene maps. We need to access the right tissue. So I can tell you right now, neuroscience is going to be the very last field to adopt these technologies, because it is so incredibly hard to get brain tissue. I'm guessing cancer is the first field that will likely be adopting these technologies, but we need the right tissue depending on the question of interest. Spatial maps, right now, they're all being created from very small sample sizes. So you may see, I have 50,000 spots, but that probably comes from 5 to 10 donors. That's not necessarily the end that we care about. We care about how many donors did these spatial replicates come from. The early days of single cell was the same. All the single cell atlases came from 1 to 2 donors or tissue sections, or tissue samples. And it's the same for spatial. Right now, we're trying to use these technologies, generate these atlases, but they're all very small sample sizes. We need larger sample sizes, and that will come with time. We very much need streamlined and computational bioinformatics tools to process these data. The tools today are really the Wild West. I'm happy to talk more about that if anybody's interested. Cost was brought up earlier today. I think about it as the following. So if we're going to spend all this money, which it is like thousands of dollars to profile these tissue samples, if we're going to spend all this money, we need evidence to convince clinicians that this will positively impact patient care. It's not just, for example, cancer. It's often diagnosed using an H&E image that a pathologist looks at. If we are going to use these technologies in the clinic, we need to convince the clinicians that this is a meaningful investment or lower the cost. And they are getting better. They are getting better. And then we also very much need basic researchers to work with clinicians to make this jump from basic research to the clinic. So the power of persuasion, in my opinion, it's going to really require a significant amount of high quality data and evidence. That's one way to do it. And then also the people who are running the assays in the clinic, they will be the ones making the decisions. They need to be very comfortable with these assays. And I think the community itself is still trying to grapple with, are these assays actually giving us the information that we want? How can we make it better? How can we gather less errors and things like that? So I think there's still a ways to go, but this would be my suggestion on how to get there. Okay, that's the end. Thank you everyone for your time. Happy to take some questions. Sure, thanks.
Thank you very much again. I have a quick question about, it was, I think, 10x Visio. What was the thickness of the fresh tissues there?
Oh, the question was, what's the thickness of the tissues? I think it was 10 microns.
I see. About a cell size, kind of. I see. And because, okay, then it's reasonable. But how was it possible to fresh cut them in 10 micrometers?
That is a question for my collaborator. I don't know that
I
answer.
see. Okay. Okay.
Yeah.
Thank you.
Sure.
Hi there, great talk. I've got a bunch of questions, but I think the one that is the most important. So if you go all the way back to your single cell section, you talked about the run times of different state-of-the-art clustering algorithms, and you mentioned that some of them take longer than your mini batch method.
Yes.
I was curious if the classification was any different between mini batch and state-of-the-art.
Oh, that's a great question. Yeah, I didn't touch on that at all. Sorry. So the classification was very similar. So we were actually asked by reviewers for this exact analysis, like, do you get different results? We put all that in the supplement. The short of it is, no, we got very similar results. But my pitch is, or I mean, one of my pitches, hopefully, is that I am, like, you as a researcher have to decide, like, your comfort with the tool, but often speed is, like, something that's crucially important. And as we begin to, like, generate these larger and larger atlases, my pitch is to develop scalable tools. And I think the researchers who originally developed these tools never really intended to have billions of cells, but now that we have them, the goal is to maybe take these methods and actually make them faster. Hopefully, like, this kind of talk would motivate them for investing energy in making their tools scalable. At the end of the day, though, the predictions or the labels that we got for the cells were very similar, and that's in the supplement.
Thank you.
Thank you. So I had a question also in the single cell area.
Sure.
You started by mentioning that single cell transcriptomic data is extremely sparse. And if I understand correctly, k-means, mini-batch k-means is kind of like data agnostic method. You can throw it at any data.
Correct.
So do you think there is a potential to squeeze even more performance by taking advantage in some way of the fact that the data is sparse? Like, is there something about sparsity in the data that can be exploited?
Great question. Oh my gosh. So I took out a whole section of my talk around sparsity of single cell and dimensionality reduction. So if I had to summarize your question, it would be, how can we leverage the power of the sparsity that we see in single cell for downstream analyses? So bulk RNA-seq and tools that assume you have some kind of Gaussian data, they, like PCA, clustering, k-means, they all assume that the data have some kind of Gaussian normality to them, but these data are incredibly sparse. And so we actually showed there are actually better models that you can use to model the error terms for these data, specifically the Poisson and the negative binomial and things like that. In another project that I'm happy to talk to you about offline, we generalized PCA to count-based data. So we took, like, PCA, which is designed for Gaussian data, and then we generalized it for Poisson data, for example. So what we do in practice is actually take GLM PCA, use that to get a set of residuals. We assume a particular model for each gene. We calculate a set of residuals. It's like a deviance residual or a Pearson residual. You take the expected count and you take the observed count and you calculate the difference. And then genes that are not that different from what you expect, they don't have much biological variation. Genes that are very different than what you expect, they have a lot of biological variation. And if you're curious, under generalized linear model theory, those residuals are Gaussian. So you can actually take those residuals and pipe them straight into PCA. And we get significantly improved results, but I felt like that was just gonna, like, be a lot for the talk, so I took that out. But I'm happy to talk to you about it offline.
Sounds really cool.
Thank you so much. I had a quick question about the, so if in the context of spatial transcriptomics, I mean, there are new technologies that are coming out with higher number of genes across multiple number of spots and high resolutions as well. But then would that mean that that could overtake single cell RNA sequencing at some point? Because, I mean, we have a lot of information with the additional dimension of space as well as time, perhaps.
Yeah. So if I had to summarize your question, is that there are newer technologies coming out that are spatial and single cell with maybe capturing thousands of genes. And
Right.
is that just going to replace single cell?
Right.
I think the jury's still out. I mean, single cell's been around long enough that people have started, have studied it and understand, what are the biases in the data. I only could guess at this point what the biases are for the image-based technologies. I have 2 students working on it in my group. So I think, yes, eventually, I could imagine that. Though I will say the technologies, at least that I'm aware of, can get up to only about 5 to 10,000 genes. And even then, it's kind of sketch. So I would say if we could get up to the full transcriptome, that would be really great. And I would be more into it. Again, it depends on your question of interest. If you don't need 10,000 genes or 20,000 genes, there's no reason to go for that, to spend the money on that. But if you don't have any idea what's going on in your tissue, you want to spend the money and get the full transcriptome. It really depends on what your question is. So single cell offers the full transcriptome, but it doesn't offer the space component. And we're really bad at trying to predict communication without that spatial component versus just seeing it very clearly. Yeah.
Thank you.
From the donors, we could get any other format other than HDF or TileDB or ZAR?
For the donors, are they available as different formats? Is that what you said?
Yeah.
So the donors that I talked about, the 3 donors that I talked about today, yeah, they are all available, freely available in HDF5 and a spatial experiment object. It's an object in Bioconnector that you can download the data in, but all the data are also in a Globus endpoint that you can download. So it's just the output that you get from Space Ranger. So Space Ranger is a tool that you can use to process the spatial transcriptomics data from 10x Genomics, and we took all of that raw output and we just made it available for someone to download.
Thank you.
Okay.
Here's-
Oh.
Thanks very much for the talk. This is a very great talk. So about this mini batch k-means, I was just wondering, would parallelization of the algorithm play a part in such algorithms, or only because you need to write the data on the disks and that is the limitation for parallelization? Also, my next question is, would there be any role for any neural network type of algorithm, deep learning type of algorithm in such research? Yeah. thank
Okay.
you.
So excited you asked that. Okay, so the first question is parallelization with mini batch k-means. Yes, so you could definitely parallelize this. We didn't, we just, I think you can parallelize this across nodes. So you just split up the batches and it does this an embarrassingly parallel way. But we
Yes.
didn't think about it any more than that. But I guess you could imagine making this even faster. And then in terms of opportunities for deep learning algorithms, yes. So for example, the field itself, I hope that you got the sense that I don't have a lot of labeled data. I don't have a lot of images where I have information of this is breast cancer, this is not breast cancer, for example. We're often we go into a new region of the brain and we're trying to discover what the patterns are in the data. So all of these results are they're almost always unsupervised, right? So if we are going to talk about deep learning, which is very exciting, it has to be kind of unsupervised flavored. It can't be supervised analyses because, again, I don't have that. So I have been reading the literature quite a bit lately in deep learning models with the goal of essentially unsupervised analyses. And the field has really skyrocketed recently in what's called self-supervised or semi-supervised analyses. So I can talk about more if you want, but this is how people are starting to use deep learning in the context of single cell and spatial. So they use these models as a way to refine the latent embeddings that you get from some kind of graph-based autoencoder using contrastive self-supervised learning. And then using these embeddings, you can do downstream analyses with them. You can identify spatial domains. You can identify cell types and things like that. So at the moment, my excitement, And the opportunity that I see most clearly for deep learning in these kinds of data are self-supervised or semi-supervised, and I'm quickly trying to teach myself as much as I can about these topics, but it is not my area of expertise, I'll be honest. But I do see a lot of people using these in the field, and I see a lot of improvement compared to methods that are not using these tools. It's just, so it's really exciting to see.
Thanks so much.
Sure.
Okay, do we have any other questions? Oh, yes.
Thank you so much for that talk. It was really great. Could you speak to how multi-omic integration is kind of playing into spatial transcriptomics? For example, do you use like bulk DNA and bulk RNA and maybe even metabolomics to, I guess, model what you're seeing with the spatially resolved transcript?
Yeah. So you want to talk about metabolomics specifically?
Everything.
Well, I will tell you, there's like a whole field that's currently trying to develop multi-omic spatial. So Rongfeng is developing DBIT-seq, which is like attack an RNA in 2D space. So there's like technological developments that people are investing a lot of energy in right now. Also, people are quickly trying to commercialize those products as quickly as they can, so basic labs can buy those products. So I see a technology come out, and then I'm imagining within 1 year's time, there's going to be like a company that's available to do that. So it's fascinating to see it in real time. So there's like technological development of two modalities in the same tissue. Then there is like a whole slew of methods that are basically taking, like let's take, I take a tissue section, and I want to map single cell data to a 2D space. So that's another type of omic that people are thinking about, where maybe the cells were site-seq, where you have protein and RNA, and then your space is just RNA, and now you suddenly have predicted protein information in 2D space. Then there are a whole slew of methods that people are developing, like assume you have some kind of cells or spatial data that are just attack, and then a different set of cells or a different set of spatial data, but from like the same tissue region of interest, and you want to integrate these. They're thinking about this as like data integration, lots and lots of development there. So yeah, it's the Wild West.
Okay, thanks. We've been getting a lot of interesting questions. I think, so I think we're wrapping up. I can thank Dr. Hicks again for a great talk, a primer plus talk.